Hello, we're glad you've joined us for this live webinar, Small Platform Enables Big Change, Nanotech-assisted discovery of novel biomarkers for disease diagnosis. I am Michelle Ashton of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration, and is made possible by an open educational grant from Agilent. Agilent has had no input in the selection of speakers, content, or mode of presentation. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Ask a Question box located on the far left of your screen. I now present today's speaker, Dr. Tony Hu, the Associate Professor of the Biodesign Institute, Arizona State University. To learn more about our speaker, please visit the Speaker tab at the top of the presentation window. Dr. Hu will now begin his presentation.
Hi, everyone. This is Tony Hu, Associate Professor at Arizona State University. So my team has been focused on the development of nanotechnology for biomarker discovery and quantification in the blood. So today we focus on the tuberculosis diagnosis using the nanoparticles. So why the TB diagnosis is so important? Because right now there's like 1/3 of the world population still got infected, and 5% of them in the high risk to develop the active TB. So in 2016, there's about 10 million people diagnosed as new case, and 1.7 to 1.8 million people died by TB. And the children is a really challenging cohort to diagnose the active TB. And so there's like, especially for the HIV positive, HIV and TB co-infected patients, and it's extremely difficult. And and the TB actually has been here for like centuries and or thousands of years. And why it is still so difficult to diagnose? And there's we can diagnose 10 million people every year, but there's about another 10 million people cannot be diagnosed accurately. So although we have the hundreds of technology on the market for TB diagnosis, but the central concept was never changed. And the doctor always rely on the isolation and the profile of bacteria from the sputum, and as the gold standard for TB diagnosis. So such methods generally need the very long-term around time, and it's usually 6 to 8 weeks to get the final result. And it's really difficult because not only because their low sensitivity and specificity, and also we have a lot of extra pulmonary infected patients, and also pediatrics. And for those patients, only relying on the sputum, the collection of the sputum is very challenging. And also recently we have some new technologies, and like a gene expert, they can really speed up the detection procedures. And the problem is they still rely on the sputum, and also the DNA profiling from the sputum cannot differentiate the alive bacterial and the dead bacterial. So they have the limited function in tracking the treatment efficacy, which is also very important for TB management. And we also have the blood test. So the blood test is highly desired to address this problem. And the current available blood test is AGRA or T-spot. This rely on the long-term memory of T cells And so the problem for those methods is they cannot differentiate the active TB from a latent TB. So it may be functional for the developed country, but for developing countries, since they have a larger population of infected patients, and the primary mission for the clinical doctor is how they can quickly or rapidly identify the active TB, the patients with active disease. So in the 2015, I have a chance to attend the conference at NIAID, and the director at NIAID, Dr. Tony Fauci, gave the presentation and listed the 3 highest priority for TB diagnosis. And the first one, can we have the rapid method which is independent of bacterial isolation, for example, like culture-free? And also the second priority is, can we have a method and not rely on the use of a sputum, and which can cover all the forms of tuberculosis, like pulmonary and extrapulmonary, and also the TB, HIV and TB co-infected adult and pediatric patients. And the third one is also very important. Can we have a method which can provide the quantification information in detecting the treatment response? Okay, and so based on such need, and we actually generated such hypothesis. At first, this is very straightforward, and we're trying to capture the antigen secreted from the tuberculosis and in the blood. So we actually spent almost a year to develop the ultra-sensitive ELISA method or immunoassay method to capture those antigens. And so even we capture them and we realize this is so difficult. And so why is it so difficult? There's 2 reasons. The reason number 1, and those biomarkers in the ultra low concentration in the blood. So the clinical doctor, they don't like to use the biomarkers at a very low concentration because they may cause the huge overlap between the disease group and also the health group, and leading to the false positive or false negative. And the reason number 2 is even you capture those antigens, and in this case, we target 2 well-known the MTB antigens. One is ESA6, the other one is CFP10. And even you capture them in the blood, and you cannot make a decision the patients are really infected by MTB or other non-tuberculosis mycobacterial, for example, like Mike Kansasi and Mike. And so in the U.S., the NTM, which is a non-tuberculosis mycobacterial, is more popular than MTB. So that means even we capture the antigens from the blood, and we cannot apply them to the clinical use. And so how to address this problem? And we actually further identify the peptide fragment in these two antigens, and which is highly specific against the MTB. So one from the CFP10 and the other one from the ESA6. So the use of a peptide can enable us not only to identify the specific antigen from the MTB, and also they can break or dissociate the bonding between those antigens with the home proteins, the host proteins, or their antibodies. So they can release more signals representing those peptides. And so we have the right markers, and the next one, we're setting up the method. Here is the place we're using our nanotechnology. So to better profile those peptides, and we have to rely on the use of mass spec. And which mass spec is the best option for the clinical application, and we select a multi-TOF mass spec. But the multi-TOF mass spec, they have several limitations, including the low sensitivity. And so how to address this? And we introduced NanoDisk to enhance the signal. And the NanoDisk is made by, it's actually the nanoporous silicon nanoparticles. And so why we want to use the nanoporous structure, because they have the enlarged surface area, which can efficiently enrich the target peptide, as shown in this figure. So at first, we have a serum, and we do the very rapid digestion in the microwave, and it's about like 20 minutes. Then we got the peptide in the solution. Then we mix the digested solution with the nanodisk. Nanodisk is conjugated with the customized the peptide antibody. So in this step, the nanopore apply the first function, which is for enrichment, and the next, without elution, and we directly spot the nanodisk conjugated with our target peptide on the multi-target. Then once the laser in the multi is shooting on those nanoparticle, and the nanopore will perform the second function, which is called energy transfer. So they absorb the energy from the laser, and in the nanopore, preserve the energy in the nanopore, and efficiently pass those energy to the peptide target, and enabling them for better ionization. So, and by using the nanoparticles, and we can dramatically increase the sensitivity for those peptide detection. And as shown in this two spectrum, one is for CIP10, and the other one is for ESA6. Okay, so the next step, and we're building up the quantification method. So we involve the isotope labeled peptide as the internal standard, and then we can build up the standard curve for absolute quantification, as shown in this spectrum. And you can tell the right curve that's representing the TB patients, and the blue curve representing the health control. So both of them is showing the peak at the location of 1603. That's the internal standard, but only the TB patients is showing the peak at 1593 for CIP10, and the 1900s for ESA6. That's for the MTB secreted antigen peptide. Okay, so compared to all other the available method on the market, and the NanoDisk mass spec method is showing the multiple advantage. The first, we can help the doctor quickly identify who has the active TB, and also the blood test can cover the different form of tuberculosis, and which is the quantifiable, and we can be used in the tracking the treatment efficacy, and also the the sample to answer time is we can shorter to the 2.5 hours, and also the cost per test is less than $10. And so, once we set up the method and we start the very preclinical validations, and first we have 17 pulmonary TB and also the 19 extrapulmonary TB. So here we can detect most case for different form of TB infected patients, and there's 96 health control, and none of them is showing the signal for the antigen peptide. So the specificity is very ideal. And we also tried the pneumonia and 40 pneumonia patients, and there's 2 patients showing the very weak signal. And so when we checked the medical record for those patients, and we found out there's one patient, their TST result is greater than the 3 centimeter. So in some countries, and their CDC, based on their CDC guideline, and these patients will be defined as the TB positive. And also we have a 24 NTM infected patients provided by the NIH clinical center, and none of them is showing any signal, which further showing the power of the use of the peptide selection. And so next, we also try to evaluate how this assay can quickly identify the active TB from the latent TB infected patients, and which is the primary mission for the clinical doctors in the developing country. So for the same cohorts with the active TB and compare with the 31 latent TB infection, and there's 4 patients showing a weak signal, and which is hurt our specificity to below the some number below the 90%. But if you read some literature, the review literature, and the assumption is there's like about 5 to 10% of latent the LTBI patients, they are actually at the early phase of active TB. So this really worthy for further study to evaluate if this assay can provide the early detection function for the latent TB and to convert to the active TB. And for HIV positive and latent TB co-infection, and we have a more false positive, and it's the same thing is worthy for the further exploration. And also for the diabetes latent TB co-infection, we also have 1 false positive. And so and after this, we actually started the kind of more professional clinical validations, and by collaborating with the local hospital, and they collected nearly 1,000 patients. And but based on the exclusion criteria, we removed 700 of them. And in the rest of the patients, there's 95 culture positive and 102 culture negative patients, and also 96 the non-TB individuals were pretty confirmed they're non-TB infected. And in the both culture positive and the negative, and we have a different number of pulmonary and extrapulmonary TB cases. The first, we like to see if this method can provide the similar sensitivity in the pulmonary and extrapulmonary TB. So as you can tell, and in the pulmonary TB, and the assay reached the sensitivity at 88%, and in the extrapulmonary TB cohort, and we reached 90% sensitivity. So the sensitivity is pretty similar in these 2 cohorts. And the next, which is a very the interesting point for the developing country doctors, and so we compared the sensitivity for this assay in the culture positive and the culture negative patients. And in the culture positive patients, we reached 92%, and in the culture negative patients, we reached 85%. So it's so our hypothesis was validated, and this result was published on the Clinical Chemistry the last year, and as the cover story, and also we move further to validate this method in tracking the treatment efficacy. So the local hospital help us to collect the patient samples and in days. And in the most patients, we can detect a rapid decline of blood antigens and for those patients. And so this is really worthy for further study and to further evaluate if they can closely track the treatment efficacy or TB recurrence. And so the next battle is against the HIV and TB co-infection, because in the field we call this cohort as the double trouble. And so we got some patients with co-infection with both pulmonary TB and extrapulmonary TB, and we reached a decent sensitivity compared to the traditional method, and also for the non-TB controls, and the specificity is 89%. And so this is very surprised to us, and we found out the blood antigen level in the HIV and TB co-infected patients is even higher than the HIV negative but TB positive patients. We explained this phenomenon as by the immunocompress. But it's still worthy to further study. And the next, we challenged the method with the triple strat, which is the pediatric cohorts. And in the first group, the very small pilot study, and we reached the sensitivity at 82% to 100% in a different group, and also 100% for the specificity. So once we report this study to NIH, and they actually sent us another cohort. And at first, I think that's another pediatric cohort, but by closing study the information, and we realized they're actually infant cohort. All the patients was recruited at 3 months old, since they have the HIV infection. And so this including the 635 infant, and so eventually there's some patients developed the active TB, and so it's perfect cohort, and also the first blended study we conducted. And recently we provide all the results to the NIH, and all information was unblended. And so we analyzed the sensitivity and the specificity for the different group. And at first, for the HIV negative group, and we reached 75% to 100% sensitivity. But for HIV positive group, we reached 96% to 100% sensitivity. And all the specificity will remain at 97 to 99%. So here, this table is showing the comparison between our study, our method with others like culture, smear, and skin test, and chest X-ray. So yeah, the method is showing a better sensitivity and specificity. And there's a special case I really like to present because it's so interesting. And this is a single case. And this child was recruited at 3 months old, and starting from the week 0, and at the week 84, he was diagnosed the TB positive, and but since his blood sample was collected every 12 weeks, so when we test his blood sample at week 36, and we already detect the signal for CIP10, so which is 50 weeks earlier than the gold standard. So which can really provide this showing the potential of this assay for early detection of active TB. So after joining the Arizona, so we further expand this study to serve the other pathogens, especially for other mycobacterial infection. So we further developed another technology, another platform to provide the multiplex detection, which is using the antigen 85B as the target, but using the single fragment based on the teeny tiny difference in the amino acid sequence, and we can differentiate the 5 major NTM, including and also TB, and in the same spectrum. And also, we also identify the specific peptide and antigen peptide against the HIV-1. Then we can right now we can profile the both HIV-1 antigen and also the MTB antigen peptide on the same spectrum. So, which is very useful for the clinical management for the patients with co-infection. And also we applied this method to serve another infection disease, and which is Ebola. So and we target on the VP40, and also select the 3 peptide, and which can differentiate the 5 strains for Ebola. And so the spectrum showing on the right side, that's provided from by our collaborator in the U.S. Army Research Institute of Infection Disease. And so we cannot conduct this study in the lab using any animal or human samples. So we just provide our protocol and to them, and they test the monkey blood they collected at their site, and so that's the spectrum. So using the peptide pattern, we can differentiate the Zaire and Sudan Ebola, and so that's all we'd like to present today. And at last, we'd really like to acknowledge the contribution from our collaborators, especially the clinicians collaborators, and from the worldwide, and also the current group member and our alumni, and also the active fundings. We have many other fundings, but they're expired. There's only space for us to show the active fundings. So now I open for the questions.
Thank you, Dr. Hu, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click on the Send button. Dr. Hu will answer as many questions as time permits. The first question is, why do we need a blood test for TB diagnosis?
Yeah, so the blood, as I present, so the traditional method or the gold standard method for TB diagnosis now is the sputum culture. They're trying to isolate the bacterial and profile them, and but the limitation is there's nearly like 50% of patients in the developing country, and you cannot identify any bacteria in their sputum, and so most of them they're the extrapulmonary tuberculosis, and the bacteria could locate it not in the pulmonary system but other organs like the brain tuberculosis or the bone tuberculosis. And for those patients, it's really difficult to identify to be diagnosed using the gold standard. And also, it's really difficult for children to collect their sputum. So the sputum test has some limitation. And the blood test can cover the most forms of subforms of tuberculosis infections, no matter your pulmonary or extrapulmonary. And the current available blood test like IGRA or T-spot, and cannot differentiate the active TB from the latent. And that's why we developed this method to directly target the antigen secreted from the bacterial in the blood, so we can provide the direct evidence for active TB infection.
Thank you. How do you compare your assay with other blood zero-based tests for TB?
Yeah, so we're actually comparing our assay with many others, and some other methods like the Gene Expert, they do show the very quick turnaround time, and also the very high sensitivity in the pulmonary infected the patients and pulmonary TB infected patients. But the shortage for the Gene Expert is difficult for them to track the treatment efficacy or treatment response, because the DNA they're profiling from the sputum cannot differentiate a live bacterial and dead bacterial. And so as we show in the slide of a treatment efficacy, the antigen level is very sensitive for the treatment. So that could be the one advantage.
Thank you. What is the advantage of using peptides as biomarkers other than the whole antigen protein?
Yeah, well, we ever used the whole protein as the biomarkers, and we developed the ultra-sensitive immunoassays to trying to profile those whole proteins. But we got two the problems, and one is those proteins, they're in the ultra-low concentrations in the blood, and so they could have the reason they probably the secretion rate, the secretion of those antigen is is at a low concentration. And another major reason is once the antigen present in the blood, they could be very rapidly encapsulated by the host protein or the bond with their antibody, and which is actually hurt the sensitivity using the immunoassay to profile the whole proteins. And by using the peptide, and as we mentioned, the selection peptide based on the database, and so we actually, those selected peptide is highly specific to MTB and not to the other, the NTM. So, and also by during the digestion step, we can also we can also dissociate the bonding between the antigen and the antibody and also other host proteins, which can release more signals representing the MTB antigen.
Okay, it looks like we have time for one more question. What is the commercialization plan for the assay?
That's a good question. We actually already have the company established in Phoenix and in 2018, and at the end of last year, we also have the state fund and from the investment and for the investment, and so right now we're working on the different milestones, and the company is preparing the Q submission, and we will set up the connection with the FDA for the approval of this assay. And also recently we also got the sponsors from the DoD, and to further push forward this assay for the clinical validation, we're going to collaborate with the company and also the clinician doctors at the Baylor College of Medicine for the multi-site the validation. And so I would say probably for the 2 years, and we may have this method to serve the clinical site.
I would like to once again thank Dr. Hu for his presentation. I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank our educational sponsor, Agilent, for today's educational webcast. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.